I'm Sean Scott, Vice President of Market Development for QIAGEN. I'm here to represent QIAGEN and Intel in terms of the collaboration between QIAGEN and Intel in genomics and bioinformatics. With the QIAGEN Sample to Insight solution, we're enabling healthcare providers to build out highly scalable, efficient sample to insight solutions. Essentially, when they generate data off of molecular profiling assays or next generation sequencing instruments, there's a tremendous amount of data that requires highly scalable analysis and interpretation. And this is where Intel and QIAGEN, working in collaboration, come to address those high throughput computing requirements that healthcare providers have relative to developing these capabilities in-house within their data centers. So what I'll mention here is that the basic steps are once a DNA or an RNA sample is collected from a particular patient, there's a level of enrichment and preparation that takes place, what we call a wet lab step, and then there's the sequencing, which generates a great amount of data that requires analysis and interpretation. What I'm going to do is highlight for you today the steps from the raw reads off of a sequencing instrument to a report out to an ordering physician. From an analysis perspective, there are three basic things that need to be done with NGS derived data. There is assembly, alignment with known human genome maps, and calling. And calling is specific to calling the specific variants or alterations that are unique to a particular patient that may have biological significance and or be causal or influential in whatever disorder and or risk predisposition the patient may have. Post-analysis, once we've identified the unique genetic markers in a particular patient, it now becomes a question of how to best interpret that information, present that information to an ordering physician, patient, and or counselor, and ensure that the report that's being provided to them with the supporting evidence is of a sufficient coverage and depth to enable them to gain better insights and make better decisions from that information. So what we have right here is a view of QIAGEN's Clinical Insight platform. This essentially presents the unique genetic markers, or what we call variants or alterations, to the individual within the molecular diagnostics laboratory for a particular patient. In this case, you will see there was 4 unique genetic markers detected that the system has determined have biological significance and or deemed actionability for a particular patient. In this case, we're looking at an oncology patient with a somatic cancer diagnoses, and we have 4 distinct markers that have been detected in that particular patient sample and thus require review by a professional within the diagnostics lab. What you will see here is a precomputed classification. Once particular genetic markers are identified in a particular patient, the first step for a lab is to understand which detected variants are real and which might be false positives. Once they've done that, they want to deem the biological significance of that particular variant in terms of pathogenicity. These classifications are really based upon guidelines provided by the professional associations such as the American College of Medical Genetics, American College of Molecular Pathology, etc. And we basically implement and codify those professional guidelines within the software-based platform so that we can enable clinical testing labs to pre-compute those classifications, whether those be biological classifications and or deemed actionability of particular variants and alterations based upon professional guidelines. So in this case, we have a BRAF mutation. It is deemed to be pathogenic. Particular lab variant scientists will review the rules upon which that classification of the detected variant was determined, and then they'll do a more thorough genetic assessment of the particular pathogenic mutation within the Clinical Insight platform. What they can review is known treatment options. These treatment options can be FDA approved for the patient-specific cancer type. They can be FDA approved in other cancer types. They can also identify any potential clinical trials for which this patient may be eligible for if there are not known FDA approved therapies for the patient's cancer type. Why this is important in terms of presenting the lab and the physician with all the available clinical trial options is we're finding in oncology, for example, that less than 3 to 5% of oncology patients are ever aware of and or enroll in clinical trials with investigational drugs where they may benefit from those treatments. And so we find that there's a tremendous opportunity here to improve the identification of relevant clinical trials for patients. Once we've done a thorough review of the targeted therapy and trial options, which are tailored to the specific cancer's diagnoses, phenotype, distillation of genetic markers that have been identified, and in some cases where a multivariate analysis is required and potentially combination therapies are required, we can then move through the workflow within a clinical testing lab to review the information that we intend to report out to the ordering physician based upon the test requisition form and the details provided by the physician in terms of the information they want, the consent of the patient in terms of the level of information they want available on their report, as well as the reporting policy of the particular lab in terms of how they report the various information out. Here you will see that we have a positive result on the interpretation. We have 3 variants and or alterations that are deemed to be biologically or clinically significant for this particular patient and or actionable, and thus we'll report out on this information. Along with each of these variants and alterations that are specific to this patient, we're providing a summary synopsis of the role of this particular variant in the disease, the relationship to treatment and trial options, whether or not there's prognosis information available, such as survival rates. And then we're enabling the labs to report out on treatment and trial options so that the physician can review all treatment and trial options and make the decisions on which combination therapies to put their particular patients on at any time. Again, all intended to be clinical decision support for the clinical testing lab, not intended to be a diagnostic tool to be used directly with physicians and or patients, but highly effective in enabling the clinical testing labs that are serving their physicians to identify all relevant information that may benefit this particular patient. With that, we're also providing the clinical testing labs within the healthcare systems with all of the available reference information, so they have traceability to why particular treatments and trials were identified and being reported out to a physician so that the physician can review the evidence supporting what is being reported out by the lab, and this is critically important. Once the individual within the laboratory has reviewed these preliminary results and is ready to sign out the report, they can simply generate a tailored report directly off the Clinical Insight platform and submit that either through a physician-facing portal or email or fax, whatever the methods are they use today. So let me just give you an example of a report. There we go. And this is just a generic reporting template that gives you an example of the type of report that can be generated off the Clinical Insight platform. So again, this one is tailored towards oncology applications, somatic cancer testing. Here we would have the logo, name, test name, etc. from the particular healthcare provider so they can brand this particular test product. We provide all the patient clinical metadata as well as specimen DNA, RNA material information that it would typically be reported out. And then everything we reviewed within the application that's being interrogated from a clinical decision support perspective, we're presenting on a physical report that a physician and or counselor can review. I'd like to speak to a little bit of the industry challenge that Intel and QIAGEN are collaborating to solve for, and that is the democratization of next-generation sequencing-based technology and the need for high-performance computing-based analysis and interpretation of that data within a healthcare setting. So while there's been great progress in improved molecular diagnostic technologies and next-generation sequencing, and institutions are doing a reasonably good job with data generation, they're fundamentally challenged with the analysis and interpretation of that type of information at scale so that they can provide those types of testing services, screening services within their healthcare system eliminating the need for them to send out for those services and the turnaround time and cost associated with that. So at the core, QIAGEN and Intel are collaborating to enable healthcare providers to develop, deploy, and scale next-generation sequencing-based technologies within their hospital systems. We first started to address the challenges faced by large healthcare providers as well as small healthcare providers in terms of deploying the basic infrastructure and workflow for the analysis and interpretation of data. And now we're working to help them deploy that through their precision medicine-based initiatives and programs to improve the services that they can provide to their ordering physicians and the patients that they serve. Work to date has been largely done on demonstrating that we can enable healthcare providers through their existing data center infrastructure to build more scalable interpretation, analysis and interpretation capabilities around NGS, and now we're carrying forward with that collaboration to identify how we can help those healthcare providers apply that to more precision medicine-based approaches.